As kindly introduced, I'm Heyun Lee from Christoph Niels Lab in German Cancer Research Center. First of all, I want to appreciate Brig again for giving me this nice chance to share my recent publication. My topic today is about the unexpected and novel function of R-Spondins as new BMP receptor antagonist in embryogenesis. So whether you are dedicating to Wnt signaling field or not, I think nowadays you may be quite familiar with the gene name R-Spondin, because among 4 R-Spondin proteins, R-Spondin 1 has been identified as a secreted stem cell growth factor and used as a critical ingredient in the organoid culture. But in addition, the other R-Spondins are also found as critical for the broad spectrum of developmental processes. For instance, R-Spondin 2 governs vertebrate limb development. R-Spondin 3 promotes angioblast and vascular development. And R-Spondin 4 is implicated in nail development. So far, all of these contributions of R-Spondins are explained based on its function as Wnt signaling agonist. So how R-Spondin can amplify Wnt signaling? Based on our long-term knowledge, in the absence of R-Spondin, the E3 ubiquitin ligase Z3 and its homologue RNF43 ubiquitinase Wnt receptor frizzled, thereby mediates endocytosis and subsequent degradation of Wnt receptor. By doing this, Wnt signaling can be reduced. But in the presence of R-Spondin, it can interact with its direct receptor LGR proteins and then sequesters E3 ubiquitin ligase Z3 RNF43 to form this ternary complex. This complex is internalized to induce the membrane clearance of Z3 and RNF43, and thereby this is how R-Spondin amplifies or potentiates Wnt signaling pathway. A recent publication implicates that now our long-term health view of R-Spondin function should be challenged and revised because, first in 2017, Yan et al. Reported that Wnt and R-Spondin show non-equivalent function in stem cell self-renewal. Moreover, in 2018, 2 individual papers reported that, different from our knowledge, LGR, which is very well known with R-Spondin receptor, can be dispensable for R-Spondin to function in several contexts, meaning that our understanding of R-Spondin should be revised from now on. So these concerns gave me 3 major questions as shown here. First, do R-Spondin possess Wnt-independent functions? Do they engage other receptors except Wnt signaling related one? If so, in which physiological process is relevant? So these findings gave me the idea of the Wnt-independent R-Spondin to function study, and I revisited our previous publication in 2004. At that time, Kazanskaya et al. Found that in Xenopus embryonic tissues, BMP4 overexpression can activate BMP direct target gene FANT2, you can see here. But when she co-expressed R-Spondin 2 together with the BMP4, this target gene expression was completely diminished, indicating that somehow R-Spondin 2 can block the BMP4 signaling and regulate the target gene expression. But at that moment, none of the mechanism was investigated, and this was just it. So I established my research aim as that denucleating R-Spondin function on suppressing BMP signaling pathway during embryogenesis. So from now on, I will show my experimental result, part 1, regarding the R-Spondin function of BMP signaling antagonism. To fully analyze whether R-Spondins can antagonize BMP signaling, I utilized HepG2 cell line and performed BMP reporter assay. As you can see here, BMP4 stimulation can increase the signaling from here to here, and interestingly, R-Spondin 2 and 3 treatment can decrease the signaling, but it was not the case for R-Spondin 1 and 4. To investigate whether this function of R-Spondin is indeed Wnt-independent phenomena, for that purpose, I knocked down beta-catenin, which is mostly well-known a transcriptional cofactor for the Wnt signaling pathway, and performed the report assay in the same setting. You can again appreciate here that R-Spondin 2 and 3 can block the Bmp signaling, but is again not the case for R-Spondin 1 and 4, indicating that, different from R-Spondin 1 and 4, 2 and 3 specifically antagonize BMP4 signaling pathway in HepG2 cell line. To fully confirm that this phenomena is indeed Wnt-independent function, this time I tried to knock down the R-Spondin receptor LGR as well as Wnt co-receptor LRP6 in the same cell line. Again, you can appreciate here that, similar to the control situation, R-Spondin can block the BMP signaling from here to here. Even it can do its own job without LGR4 and 5, also without LRP5 and 6, clearly meaning that R-Spondin 2 and 3 antagonism for BMP signaling is independent of Wnt signaling pathway. This is quite important finding because so far, as I mentioned in the introduction part, every kind of study only focus on the R-Spondin function on the Wnt, but now we found that R-Spondin can do another job which is independent of Wnt signaling pathway. Next, I asked which domain of R-Spondin is required for BMP inhibition. It is very well known that R-Spondin share crucial main domains. For example, as shown here, it is consistent with a signal peptide and the N-terminus, and it has two furin 1 and furin 2 domains, which is known as important for the Wnt signaling. At the C-terminus, there is a TSP1 domain reside, which is known to be important for the Wnt PCP signaling pathway, which can also interact with the heparin sulfate proteoglycan, such as syndecan-4. But I want to mention here that TSP1 domain importance is not well studied in the context of the Wnt beta-catenin signaling pathway. Based on this information, I generated the deletion mutant form of R-Spondin 2, for example, delta TSP and delta furin 1 and 2, and then performed the BMP report assay again to check which domain of R-Spondin is important for the BMP signaling pathway regulation. Again, you can see here that BMP4 signaling can be decreased well by wild-type R-Spondin 2. But when I treated R-Spondin 2 delta TSP1 or R-Spondin 2 delta furin 1 and 2 form, you can see that now it cannot block the BMP signaling anymore, indicating that both TSP1 domain and furin domains are important for BMP4 signaling inhibition. Moreover, we want to check by loss of function approach to check whether R-Spondin 2 is indeed important for the BMP4 signaling. So my colleague, Dr. Soon, performed the post-SMAD1 Western blot, and post-SMAD1 is the hallmark of the BMP4 signaling activation. He utilized H5081 cell line and knocked down R-Spondin 2 and checked the sensitivity of the cells to BMP4 signaling. You can see here that 2 hours of BMP4 stimulation can increase the post-SMAD1 level as 1.1-fold. But if there's no R-Spondin 2 in this cell line, interestingly, almost 2-fold induction of post-SMAD1 occurred, meaning that knockdown of R-Spondin 2 sensitized cells to BMP4 signaling independently of the Wnt signaling pathway. Because knockdown of LRP5 and 6 doesn't do such of a strong fold induction like R-Spondin 2 knockdown. So again, clearly says that our finding is Wnt-independent function of R-Spondin 2. So as a summary of part 1, we clearly say here that R-Spondin 2 and 3 can act as both Wnt agonist and BMP antagonist. So next I asked, what is the physiological role of R-Spondin BMP antagonism? And this question led me to the result part 2. So here I will briefly remind or introduce you about the function of Bmp signaling for the dorsoventral axial patterning in xenopus development. This cartoon represents the simplified regulation mode for the dorsoventral patterning regulated by Bmp signaling pathway. In the gastrula embryo, in the ventral part, there's Bmp ligands such as Bmp4, 2, and 7 are expressed. In the dorsal part of the gastrula embryo, Bmp antagonists such as codinogin and twisted gastrulation is expressed. And therefore, the Bmp morphogen gradient is formed along ventral to dorsal, so strong Bmp signaling is maintained on the ventral, and the lower Bmp signaling should be maintained in the dorsal. This gradient is maintained until neurula and tadpole stage. That's how xenopus tadpole can form normal dorsoventral axis. But if this morphogen gradient is broken by knockdown of Bmp antagonist, for example, you can see that now this gradient is broken, then tadpole can eventually show the ventralized defect. You can see here that the smaller head structure and enlarged ventral structure of the tadpole embryo. So this is the most well-known function of Bmp signaling in dorsoventral patterning during xenopus embryogenesis. So based on this knowledge, I next try to check whether R-Spondin 2 indeed regulate BMP4 signaling in xenopus development as an in vivo evidence. So first, for that purpose, I overexpressed BMP4 mRNA with or without R-Spondin 2 mRNA, and then culture the embryo until the tadpole stage and check the defect. You can see here that at stage 32, the control embryo shows totally normal tadpole structure. But if I overexpress BMP4 mRNA, you can see smaller head and enlarged ventral structure as I introduced before. Interestingly, if I overexpress R-Spondin 2 wild-type mRNA together with BMP4, now the defective embryo is fully recovered, almost the same as normal. But different from wild-type overexpression, mutant form of R-Spondin 2 mRNA cannot rescue this defect, indicating again, as I previously shown, all of this furin and TSP domain is important to antagonize BMP4 signaling pathway. So again, this result says that indeed in vivo, R-Spondin blocks BMP4 signaling pathway. To confirm this in vivo evidence by the approach of loss of function, now I injected morpholino, which is commonly used a knockdown reagent in frog assays. So by injecting R-Spondin morpholino, I can induce the knockdown of R-Spondin 2 during xenopus embryogenesis. And then I checked BMP target gene VANT2 expression at neurula stage. So here shows the result. You can see here that when I injected R-Spondin to morpholino, so this embryo doesn't have R-Spondin 2. You can see that increase of BMP target gene expression, meaning that loss of R-Spondin 2 increase BMP signaling pathway. To check whether this phenomena is again Wnt-independent function, now I injected LRP6 morpholino. With LRP6 is again a Wnt co-receptor. So again, you can see that R-Spondin to morpholino injection still maintain the increase of VANT2 expression even without LRP6, indicating again here that R-Spondin 2 blocks BMP signaling, and this is again independent of Wnt signaling pathway. To confirm this finding again in the loss of function approach, now I analyzed the BMP target gene expression VANT1 and sizzled by in situ hybridization. So you can see here the strong purplish color, and this indicates the VANT1 or sizzled expression in gastrula xenopus embryo. First, you can see here that when I knock down BMP4 in xenopus embryo, this, of course, direct target gene expression is completely diminished. Interestingly, I found that when I co-knock down R-Spondin 2 by morpholino, this can recover or restore the Band1 and scissors expression, meaning that loss of BMP4 and R-Spondin 2 neutralize each other in BMP target gene expression, also again confirming that R-Spondin blocks BMP4 signaling pathway. This was even further confirmed by knockout approach by CRISPR-Cas analysis. To make a CRISPR of R-Spondin 2, we generated guide RNA and co-inject with Cas9 protein in xenopus stage 1 embryo, and then cultured them until tadpole stage and analyzed the phenotype. First, you can see here that R-Spondin 2 knockout by CRISPR-Cas approach shows very mild phenotype. And also, we found that the codeine, which is well-known BMP-secreted antagonist, this codeine knockout also only mediated very mild defect. But interestingly, we co-knockout or mediate double knockout of R-Spondin 2 and BMP antagonist codeine. Now you can clearly see that head is indeed dramatically decreased. And also, you can see enlarged ventral structure, which is a typical hyperactivated BMP phenotype, indicating that double knockout of R-Spondin 2 and BMP antagonist codeine synergize and yield strong ventralization, and again confirming that R-Spondin 2 indeed function as a BMP signaling regulator. To get more insight or more detailed mode of action of R-Spondin 2 for the BMP4 signaling regulation, now we analyze the R-Spondin 2 expression pattern during xenopus embryogenesis. Here you can see the published color indicating R-Spondin 2 expression during embryogenesis, and we found that this expression seems very familiar or similar to BMP4 expression pattern. So we say that here that R-Spondin 2 is co-expressed with BMP4 in xenopus embryogenesis. And this overlapping expression pattern gave us the idea or concept of the BMP SYN expression group, which we established and published in 2004. As indicated with these blue colors in this panel, so to say, the BMP SYN expression group consists of at least 8 members in BMP signaling pathway. And overall, they show the overlapping expression pattern with the BMP4. And interestingly and importantly, they found that these BMP4 SYN expression group genes encode feedback components of BMP signaling cascade in xenopus embryogenesis. So I also ask at this moment, so can R-Spondin 2 be a new member in this BMP4 signaling expression group? Because to me, R-Spondin 2 also shows all of these similar phenomena or character. To test this idea, now I injected a stage 3 embryos with the BMP4 mRNA to activate the BMP signaling, and then dissected these animal pole kept tissues at stage 8, and cultured them with or without cyclohexamide and analyzed gene expressions at stage 10. Here first you can see again the published color indicates either R-Spondin 2 expression or the direct non-BMP target gene sizzled. You can see here that interestingly, when I overexpress BMP4 in these animal kept tissues, R-Spondin 2 expression is strongly induced, similar to the BMP direct target gene sizzled, indicating here that BMP4 regulates R-Spondin 2 expression. Moreover, interesting point here is that when I culture the tissues with cyclohexamide, I found that this induced expression of R-Spondin 2 was diminished, different from direct target gene sizzled. Here we can say that, but this BMP4 can regulate R-Spondin 2 in an indirect manner, different from direct target gene sizzled. So based on this finding, we found that we conclude here that in xenopus gastrula embryo, R-Spondin shows a co-expression with BMP4. different from well-known BMP antagonists codeine and nogaine. How R-Spondin or BMP regulate the signaling? BMP4 induced the expression of R-Spondin 2, and by then R-Spondin 2 as a feedback negative regulator can block the BMP4 signaling, maybe via Vent and MXX1, because we found that R-Spondin is indirect target of BMP4. And this is how the embryo can achieve the proper dorsal-ventral patterning during xenopus embryogenesis. So far, in vitro and in vivo, it's crystal clear that R-Spondin 2 or maybe 3 blocks the BMP4 signaling pathway. So it was quite natural for me to investigate the detailed mechanism behind this phenomenon. So now let's move on to the third part of the result regarding the mechanism. So here shows a very brief overview of the BMP signaling. You can see here that there are a subset of BMP ligands which can interact with the type 1 receptor and type 2 receptor, and then it can mediate the serial phosphorylation of the SMAD protein, and eventually can mediate the target gene activation. And also there are known a subset of BMP secreted antagonists. So based on the character of R-Spondin as a secreted protein, I hypothesized that R-Spondin may regulate the BMP receptor. To check this idea, I performed the BMP reporter assay in HepG2 cell using the constitutively active BMP receptor type 1. First, you can see here that when I transfected constitutively active form of BMP receptor 1A, BMP signaling is activated from here to here. Interestingly, again, R-Spondin 2 and 3 can mediate the decrease of this signaling pathway. Not the case for the R-Spondin 1 and 4. Different from BMP receptor 1A, activin receptor 1A or activating receptor 1 or BMP receptor 1B, constitutively active form, signaling cannot be blocked by all of the R-Spondins, indicating that R-Spondin 2 and 3 specifically regulate or antagonize BMP receptor type 1A signaling pathway. Moreover, we further found that indeed R-Spondin 2 and 3 can directly interact with the BMP receptor 1A extracellular domain based on our in vitro binding assay. You can again appreciate here that R-Spondin 2 and 3 showed a strong binding between BMP receptor 1A extracellular domain. It was not the case again for R-Spondin 1 and 4 or the other signaling ligand FGF. Furthermore, we found that the KD value of R-Spondin and BMP receptor 1A extracellular domain is around 4.8 nanomolar, which is the similar range with non-R-Spondin 2 and LGR interaction. This finding was kind of a milestone for our storyline because this in vitro binding evidence and KD evidence strongly suggest that indeed R-Spondin interacts with the BMP receptor 1A, and this indeed the very first evidence showing that R-Spondin can interact more in addition to the LGRs. So we found a new receptor partner for the R-Spondin. Then we asked which domain of R-Spondin is interacting with BMP receptor 1A. To give the answer for this question, we performed a cell surface binding assay. So this purplish color indicates the surface interaction between each R-Spondin form and BMP receptor 1A. Here you can appreciate that R-Spondin 2 wild type can interact with BMP receptor 1A. Also, R-Spondin 2 delta furin 1 and 2 interacts with BMP receptor 1A. But R-Spondin 2 delta TSP cannot interact with BMP receptor 1A, indicating again that TSP domain is important for the receptor interaction. And this was the same case for the R-Spondin 3 Again, you can see that deletion of TSP domain in R-Spondin 3 diminished the binding between R-Spondin 3 and BMP receptor 1A, again suggesting the importance of TSP1 domain for the receptor interaction. To confirm again clearly the importance of TSP1 domain, we generated the TSP1 R-Spondin 1 form, and the construct which only has R-Spondin 2 TSP1 domain, and performed the in vitro binding assay. So you can see again here that R-Spondin 1 TSP1 domain cannot interact with the BMP receptor 1A, but R-Spondin 2 TSP1 domain can strongly interact with BMP receptor 1A, indicating and confirming again TSP1 domain is indeed interact directly with BMP receptor. Moreover, we try to make a chimera which has the TSP1 domain from R-Spondin 2, but still has the furin 1 and furin 2 domain from R-Spondin 1. So the purpose of this was that we want to check indeed if TSP1 domain is important. If we exchange the TSP1 domain of R-Spondin 1 into the TSP1 domain of R-Spondin 2, maybe this chimera R-Spondin 1 can act as or behave as a R-Spondin 2 wild type in terms of BMP4 signaling inhibition. So using this generated chimera, we perform the BMP report assay in healthy 2 cell line. Again, you can see here that BMP4 signaling is increased upon the stimulation. And again, as I shown before, R-Spondin 1 wild type cannot affect the BMP4 signaling pathway. But this chimera, which has TSP1 domain come from R-Spondin 2, now can block the BMP4 signaling as R-Spondin 2 wild type, clearly saying that the TSP1 domain swapping of R-Spondin 1 mimics R-Spondin 2 function of BMP signaling, and again confirming the importance of TSP1 domain. To fully confirm the importance of this domain in vivo, now I designed and generated the R-Spondin 2 delta TSP morpholino. This morpholino can make the truncated TSP1 truncated R-Spondin 2 in xenopus embryo. So if I inject this R-Spondin delta TSP morpholino, embryo only harbors this delta TSP, the truncated R-Spondin 2, so we can directly check whether this TSP is really important for the embryogenesis in terms of BMP signaling pathway. So first, you can see here that control embryo shows perfectly normal shape or phenotype of the embryo. And when I inject this R-Spondin 2 delta TSP morpholino, you can see that again the smaller size of the head as well as enlarged structure of the ventral, indicating that indeed this morpholino injection can hyperactivate BMP signaling pathway. More interestingly, when I co-inject BMP receptor 1A dominant negative form together with this morpholino, this phenotype can be restored, indicating that this phenomena is indeed comes by misregulation of the BMP receptor 1 signaling. And further confirmed, this phenomena was further confirmed by in situ hybridization. You can see here that the BF1, which marks the anterior part of the structure of the embryo, this published staining by BF1 probe was decreased nicely by delta TSP1 morpholino, confirming the shrinked head structure. And this shrinked expression is fully recovered by BMP receptor 1A dominant negative mRNA. More importantly, when I check the CISLD, which is well-known direct target gene of the BMP, you can see that it was dramatically increased in the ventral side of the embryo by this delta TSP morpholino. And this was fully restored back to normal level by injection of BMP receptor 1A dominant negative form, confirming here again that TSP1 truncated R-Spondin 2 in xenopus embryogenesis enhances BMP4 and BMP receptor 1A signaling pathway. So to sum up the result part 3, here suggests that we found R-Spondin 2 and 3 can interact with the extracellular part of the BMP receptor 1A via TSP1 domain. And this can block the Bmp signaling pathway in vitro and in vivo. Then we asked what is the consequence of this R-Spondin and BMP1A interaction. So we further checked the details of this mechanism behind this binding. To check how what is the consequence of R-Spondin and BMP receptor 1A interaction, we're going back to the xenopus embryogenesis. And we hypothesized first here that maybe R-Spondin can regulate the surface protein level of BMP receptor 1A. To confirm this idea, in xenopus embryo, we injected BMP receptor 1A EYFP tag and also as a lineage tracer membrane RNP mRNA together with BMP and R-Spondin 2 mRNA. At stage 9, we dissected the animal pool tissues and performed immunofluorescence to check the surface BMP receptor 1A level. Here you can see here the control situation. You can see very yellow lining alongside the embryonic cell, indicating that most of the BMP receptor 1A is localized nicely at the cell surface in the control situation. But when we overexpress R-Spondin 2 mRNA, now you can see that green surface signal is totally gone, so you can only see the red signals here, indicating that R-Spondin 2 overexpression reduces or decreases surface BMP receptor 1A levels. This was not the case when we overexpress delta furin 1 and 2 or delta TSP form of R-Spondin 2. You can still see that BMP receptor 1A is nicely localized and stabilized at the cell surface, indicating that R-Spondin 2 reduces surface BMP receptor 1A, and again, this function requires both furin and TSP1 domain. This immunofluorescence result was further confirmed by Western blot analysis. So my colleague Karina performed the Western blot using the xenopus embryo rhizate. Again, you can appreciate here that BMP receptor 1A protein level was dramatically decreased by wild-type R-Spondin 2 overexpression, but it was not the case for the delta furin or delta TSP1. Indicating that R-Spondin 2 decreased BMP1A level or R-Spondin 2 may mediate BMP receptor 1A degradation in xenopus embryogenesis. So far, this evidence comes from the gain-of-function approach. So next, I performed the loss-of-function approach analysis using the same experimental scheme. In this case, I injected R-Spondin 2 morpholino at the ventral side of the embryo and dissected ventrolateral marginal joint tissues at gastrula stages, and then performed the immunofluorescences to again check the BMP receptor 1A at the cell surface of the xenopus embryo. You can see here that in control situation, barely see the BMP receptor 1A at the cell surface. But if there's no R-Spondin 2 in this cell, you can see the clear increase or stabilized BMP receptor 1A. And it was the same when I injected R-Spondin 2 delta TSP morpholino, indicating that impaired R-Spondin 2 expression stabilizes surface BMP receptor 1A levels in xenopus embryogenesis. Again, this result was confirmed by cell line as well as xenopus embryo. You can see here that H5081 cells, loss of R-Spondin 2 by siRNA, increase or stabilize the BMP receptor 1A protein level comparing to the control. And again, in Xenopus embryo, when I injected control morpholino, only this level of BMP receptor 1A was detected. But if there's the knockdown of R-Spondin 2, or if there's impaired R-Spondin 2, you can see the clearly stabilized BMP receptor 1A protein in Xenopus embryo. So overall, here we can conclude and say that R-Spondin 2 destabilizes cell surface BMP receptor 1A in vivo and in vitro. The next question was quite natural for us. What is the details? How can R-Spondin destabilize the cell surface BMP receptor 1A? And then this goes to the final part of my experimental results. Coming back to the required domain of R-Spondin 2 to block the BMP4 signaling pathway, we analyzed further among furin 1 and furin 2, which domain of furin is really important for the R-Spondin 2 function to block the BMP signaling pathway. So we utilized the furin 1 deletion form of R-Spondin 2 and furin 2 deletion form of R-Spondin 2 and checked the BMP reporter activity again in HepG2 cell line. Here you can again appreciate that induced BMP4 signaling was nicely reduced by wild-type R-Spondin 2 treatment. But if I treated R-Spondin 2 delta furin 1 conditioned media, this cannot block the BMP signaling anymore, suggesting that furin 1 domain is required to antagonize BMP signaling pathway. But deletion of furin 2 doesn't do anything in terms of blocking the BMP signaling pathway. Again, you can see here that similar to the wild-type R-Spondin 2, still delta furin 2 R-Spondin can block the BMP signaling, different from furin 1 deletion R-Spondin 2. Indicating here clearly that furin 1, not furin 2, is required to antagonize the BMP4 signaling pathway in HepG2 cell line. Then why this is important, or why this gave us the insight for the future details of the mechanism? Because, as I previously introduced, it is already quite well known that the furin 1 domain is interacting with the ZM3 and RNF43, which is well-known E3 ubiquitin ligase important to block the Wnt signaling pathway. And furin 2 is known to interact with the LGR 4, 5, and 6, which is well-known R-Spondin receptor. But as I previously shown, LGRs are dispensable for the BMP signaling inhibition by R-Spondin. So we only focus on this furin 1 and ZM3 RNF43 interaction. So I hypothesized that maybe ZM3 and RNF43 can be involved to antagonize the BMP4 signaling pathway by R-Spondin 2 and 3. To test this idea, I performed the BMP report assay again. And again, in control situation, you can appreciate here that R-Spondin 2 and 3 block the BMP signaling pathway, not the case for the R-Spondin 1 and 4. But when I knocked down ZM3 and RNF43, now I found that R-Spondin 2 and 3 lost its ability to block the BMP4 signaling pathway. This is really suggesting that R-Spondin 2 and 3 require ZM3 and RNF43 to antagonize BMP signaling pathway, and giving us the insight for the detailed mode of action of R-Spondin 2 and 3 for blocking the BMP signaling pathway. To further confirm this important finding in Xenopus embryo, we again performed the immunofluorescence. So you can now see here clearly that, again, R-Spondin 2 overexpression can decrease the surface level of BMP receptor 1A comparing to the control. But when I co-injected a delta link form of ZM3, which is known as a dominant negative form of ZM3, now this reduced BMP receptor 1A surface level by R-Spondin 2 is fully restored, again indicating that R-Spondin 2 requires ZM3 to regulate surface BMP receptor 1A levels both in vivo and in vitro. Moreover, to have an idea how R-Spondin requires or why R-Spondin requires ZM3 and RNF43 to block the Bmp receptor signaling pathway, we perform the in vitro binding assay to check whether R-Spondin can make the ternary complex between ZM3, R-Spondin, and Bmp receptor 1A. So here you can see that in a control situation, ZM3 and Bmp receptor 1A doesn't interact each other. If we gave R-Spondin 1 between them, they still cannot interact. But if there's a R-Spondin 2, now ZM3 and Bmp receptor 1A shows nice interaction, indicating that R-Spondin 2 bridges Bmp receptor 1A and ZM3 in the cell surface. Furthermore, we found that this ternary complex can be internalized later on because when I checked the immunofluorescence by R-Spondin 2 HRP tagged condition media treatment, and when I checked with ZM3 and Bmp receptor 1A in the cell line, I found that internalized puncta for the indicating the ternary complex between R-Spondin, ZM3, and Bmp receptor 1A, meaning that they first form the ternary complex at the cell surface and then internalize inside of the cell. And then furthermore, we want to investigate what is the meaning of ZM3 involvement in this context. So to give the answer for that, in the H-field 81 cell line, first I check again the Bmp receptor 1A and ZM3 correlation in this cell line. You can see that in control situation, Bmp receptor 1A and ZM3 is nicely correlated by endogenous R-Spondin 2. But if there's no R-Spondin 2, now you can see that the green signal, which indicates the Bmp receptor 1A. So Bmp receptor 1A is now sustained at the cell surface and barely show the correlation between ZM3, indicating that R-Spondin is important to induce the Bmp receptor 1A and ZM3 correlation and mediate the endocytosis. Interestingly and importantly, this phenomena is totally different in case when I knock down LIP6 and LGR4 and 5. Even without LIP6 and LGR4 and 5, still ZM3 and Bmp receptor 1A is nicely correlated and internalized in the cytosolic area, totally different from Assay R-Spondin 2 case, indicating again that R-Spondin function is independent of the Wnt signaling pathway regulation. So here we can conclude that R-Spondin triggers Bmp receptor clearance from the cell surface in concert with ZM3. Moreover, further evidence showing that R-Spondin 2 leads Bmp receptor 1A to endocytosis for the lysosomal degradation, because when I check the Bmp receptor 1A localization with and co-stain with the EEA1, which is well-known endocytic vesicle marker, I found that in the Assay control treated cell, Bmp receptor nicely goes to the EEA1 positive puncta, indicating that Bmp receptor is endocytosis. But if there's no R-Spondin 2, now Bmp receptor 1A lost its ability to go to the EEA1 positive puncta, indicating that R-Spondin 2 is required to mediate the Bmp receptor 1A for endocytosis. Furthermore, when I check the Bmp receptor 1A localization with the LAMP1, which is well-known lysosomal marker, again the similar result was obtained. Bmp receptor 1A is again correlated nicely with the LAMP1, indicating that Bmp receptor 1A goes to the lysosomal degradation eventually. But if there is the loss of R-Spondin 2, Bmp receptor 1A doesn't correlate with the LAMP1, indicating that R-Spondin 2 first lead Bmp receptor 1A to endocytosis and subsequently this goes to the lysosomal degradation. And this is why we showed the decreased surface level of BMP receptor 1A in culture cell as well as embryogenesis. So a conclusion here says that in xenopus development, we found that R-Spondin is the negative feedback regulator by BMP4. So R-Spondin, we found that is the new Bmp signaling expression group gene. So BMP4 can regulate the R-Spondin expression. In turn, R-Spondin blocks the BMP4 signaling pathway during xenopus embryogenesis because this is important for the proper dorsoventral patterning. And how can R-Spondin regulate BMP4 signaling? We further found the detailed mode of action, and it suggests that R-Spondin 2 can mediate the BMP receptor 1A and ZNR3 in complex formation, independent from the well-known LGL-dependent Wnt regulation. And these can be internalized and goes to the lysosomal degradation to regulate the BMP receptor 1A protein level at the cell surface. That's how R-Spondin regulates and antagonize the BMP4, BMP receptor 1A signaling pathway. So I want to wrap up my talk with suggesting the significance of my study. So I said the three parts of the importance and significance. First, for the BMP biology, this study can suggest a new class of BMP receptor 1A antagonist. So R-Spondin will suggest that this is the very first type 1 BMP receptor antagonist by a novel mechanism. So far, no BMP antagonist specifically for the type 1 BMP receptor have been studied, but we are now suggesting that R-Spondin can do the job. For the Wnt signaling biology, as I emphasized several times, we found that R-Spondin do more than Wnt. This is totally unexpected finding. So R-Spondins possess a new role independently of exclusively studied Wnt agonism. We suggest that R-Spondin do more than Wnt agonism, BMP antagonism. And maybe the other researchers outside the xenopus development and Wnt field might be interested in this phenomena because our study can be a source of new insight for stem cell and disease. So we try to revise the rational use of R-Spondins in stem cell and cancer biology because so far most of the studies based on the Wnt regulation by R-Spondin, but now we should change our point of view. R-Spondin and BMP regulation might also be important for the stem cell and disease. So thank you for your attention. I want to acknowledge the Karina Sardel, the PhD student who worked with me, and also Dr. Stoon performed some in vitro analysis, and also Dr. Glinka, who performed the cloning and as product generation for our study, and also our PI, Christoph Niers, for the whole establishment of the study. This study was funded by DFG SFB 1324 Consortium, and we are working for the new PhD students, so if you're interested, please feel free to contact me. Thank you for your attention again, and I would appreciate your comments and questions.